The overall goal of this twin-spot MARCM protocol is to conduct high-resolution cell lineage analyses and better gene function studies.
Brain tissue is comprised of a vast number of diverse neurons. It's challenging to study the mechanism underlying how neurons assemble together during development. The main advantage of this technique is to label neurons derived from common neuron progenitor in two distinct colors, which permit the retrieval of the birth of the information of individual neurons. Therefore, this technique can help researchers to explore the cellular and molecular mechanism underlying neuronal development. Demonstrating the procedure today will be Pei-Chi, a technician from Professor Yu's laboratory.
Begin by combining twin-spot MARCM-ready males and females in a fly food vial with fresh yeast in the vial. Maintain the crossed twin-spot MARCM-ready flies at 25 degrees Celsius for 2 days to enhance the chance of mating before collecting fertilized eggs. To avoid overcrowding when the larvae hatch, tap down the crossed twin-spot MARCM-ready flies and transfer into freshly yeasted vials. Culture the twin-spot MARCM offspring until they reach the desired stage of development. Here, flies at the larval stage will be heat-shocked to induce flip-base expression. Transfer the fly vials to a 37 degrees Celsius water bath for 10, 20, 30, 40, and 50 minutes to determine the optimal heat shock for flip-base expression.